this.
Oh, oh yeah.
Please send her back.
I will. I'm gonna try something quickly. Let's see if I can pull it.
Mhm. Should I be changing the view of the? You're wondering where I am. Uh-oh. I'll go shut the door. No, I'll leave it. Leave it. It's okay.
Can you hear me?
Hmm.
I'm gonna try something. Can you hear this? Can you hear me?
I hear. I hear people. Yeah. I hope you can hear me.
All right, so I'm gonna go ahead and get started with a quick introduction. All right, everyone. So good morning. So today we have the pleasure of having Dr. Aseem Ansari, currently a member of the St. Jude faculty, chair of the Chemical Biology and Therapeutics Division. As far as his academic upbringing, he received his Bachelor of Science at St. Xavier's College and Bombay University in Bombay, India. He went on to do his PhD in Northwestern University and then did a postdoctoral fellowship at Harvard University. His research interests are varied. He has over 80 publications. His research consists of synthetic gene regulators as precision therapeutics for personal medicine, artificial transcription factors to regulate cell-state programs and enable regenerative medicine, specificity and energy landscape for synthetic gene regulator design, genome mutation, precision medicine, also chemical genetic dissection of RNA polymerase 2 transcription cycle. With that introduction, I will turn it over to our guest lecturer.
Well, it's a pleasure to be here. I've always had wonderful people join me on lab from Puerto Rico. Last one was Aisha. And I fondly remember my visit. And so, all the talented students and colleagues at UPR, I'm delighted to be in touch with you guys and be able to present some of the work that I'm going to talk about, but also encourage you guys to consider coming to St. Jude to try some of this work. And John Young Jose here, if possible. Today, what I'm going to do is I'm going to tell you a little bit about how small molecules. So, we're talking about chemical biology in the frontier currently. I'm going to spend some time talking about how small molecules have been used in therapeutics. I hope you can see my slides.
Yes, we can see them.
Perfect. So this is what I would like to go over. Basically, talking about how do you target the genome. Most of the drugs that you hear about are targeting proteins, or proteins that actually do something that will eventually be essential for the cell, or might be involved in causing the disease, or the biological outcome that you're interested in. But few people ever think about targeting the genome or being able to control genes directly by targeting the genome. And yet, um, some of the most effective drugs are those that target the genome. So I'd like to touch upon the literature a little bit, what kind of drugs have been developed over the years for the genome. So that's the first couple sections of the first half. Then we go into how have people started to think about making drugs that can go and turn on specific sites in the genome, and what might that mean, and what are the challenges that we currently face, right? The second half is a little more general, still tied to genome targeting and gene regulation, but from the perspective of this new principle that is coming up. In the past, drugs used to be small, not even past, even currently, some of the most of the focus is taken up by drugs that have certain properties, certain size, often, called the rule of five, first defined by Lipinski, right? But now the world is moving very rapidly towards things that weren't considered drug-like in the past. but are required to be able to bring biological processes together. And that principle of induced proximity, bringing things together, proximity that you're inducing with a small molecule, has become really popular because it is effective in driving what used to be considered difficult biological processes or targeting what used to be considered undruggable um, proteins and processes. And then we'll come back and say, how has that been used to target the genome and to control its function? How do you turn the gene on in a targeted way? How do you switch off a gene in a targeted way? And going even beyond single genes, how can you use these sorts of principles of induced proximity with using small molecules to do complete rewiring of the cell so that a skin cell becomes a heart cell or becomes an induced pluripotent cell that has many different things that it could turn into, and all using small molecules, all bringing different kinases and targets together. Okay, so that is the general outline. And I'll go quickly through some parts that are well known, and I'll go slowly through others. If there are questions, because I know it's a lot of material and I'm speaking very fast and I'm doing this remotely. If there are questions, please stop and ask me. It doesn't matter what I'm talking about, just stop me and ask me those questions so that we can have a dialogue almost rather than a lecture. All right. So with that, I'll start with this slide. Here you can see a lot of small molecules, and if you look carefully at their names, things like cyclophosphamide, chlorambucil, if you just go down, trabectedine, daunorubicin, gemcitabine, oxaliplatin, all these things, and even cisplatin, right? Widely used chemotherapeutics. These are very powerful and popular, not popular, I shouldn't say popular, but potent drugs that are used for these types of cancers. Look at the range of cancers that these things are being used for, in addition to things like alopecia and vitiligo, psoriasis, that's psoralen right there. Quite a few of these things have been used in cancer. And the reason they're used in cancer is because this is a place where you can take a drug like this, where is cisplatin right there, and treat a cell. And what cisplatin will do is will form intra-strand crosslinks between two guanines. You can't do this to normal cells because the cells then have to deal with the DNA damage. But in a cancer cell, you can treat them with these class of very potent and to some degree quite toxic drugs. What you're really doing is you're targeting the genome, right? And what are the principles that people use to look at genome targeting molecules? Right? These are the principles that people have been trying to leverage because what you have in DNA are base pairs, phosphates, and a certain amount of hydrogen bonding that you can leverage. Most often, these sorts of drugs that are used or to target the genome are intercalators, things that can get between two base pairs, minor groove binders, etc. So we'll go through all of these. We won't talk about drugs that are incorporated by enzymes into DNA or RNA. That's a different class of drugs, also targeting the genome, but it is synthesis in this case, right? Or drugs that can form covalent adapts like cisplatin. We'll talk about mostly the kinds of molecules that have been used. Um, you able to see my slides or?
Yes.
Okay. Um, that have been used to now develop more and more selective molecules that won't be as toxic. So we're heading towards that. So the first class, of course, are things that can get between base steps. So intercalators. And here's a whole class of intercalators that are popularly used, widely used. And some of these, this in here, anthracenes, are used for cancers quite a bit, right? Currently, standard of care. The other class are minor groove binders, things that can sit in the minor groove between the two phosphates, right there. And some of those molecules are here. And pentamidine is used quite widely. It's basically two aryl rings spaced by different types of linkers in between furans and alpha. So you'll have those sit in the minor groove like so, and different derivatives of it. So you have these two basic modalities, because if you use phosphates to bind, and you do to get affinity, Those phosphates are everywhere in the genome. So how do you control which phosphates will confer specificity as opposed to just general affinity for DNA, right? So looking at these two classes, people have focused on minor groove binders because they require engagement of the base edges. So here's a base step right there. I hope you can see my um, pointer. Others, I'll try and get you. Um, is it here? No, there. I'll try to use the pointer here. Ah, good. I hope this is clearer.
that we can see.
Here are the base edges. That's one base step. There's another one facing away from us. Here are some, you can sort of see these out here. So these molecules will sit in the minor groove, which is deep, and can read the edges of base pairs and use sugars to essentially make van der Waals contacts. So starting with this idea, people started building molecules that can bind specific sequences. How do you make them so that in the entire genome, they can find a certain combination of bases and stay there longer than they would anywhere else? And if they stay there longer, they can interfere with just this region and not other regions of the genome. So maybe reduce some of the toxicity um, while gaining the benefit of stopping something that might be causing the disease, or starting something whose absence, the gene or product, whose absence may be part of the disease. And I'll tell you the example, the latter example, much later. How do you turn something on? Turning something off, just put a roadblock and the enzyme polymerase cannot go through. But to turn something else on, you have to bring the enzyme or the polymerase there. Those are much more challenging, and that's where induced proximity has turned out to be an important um, path or strategy to achieve it, achieve selectivity and be able to turn genes on. But how do you make things sequence selective? So let's start with how the history, how it started. So start from the beginning, if you will, and see how people over the years have tried to add their insights and make chemical modifications to be able to target things in a selective way and control genes and genomes in a targeted way. And then what are the challenges that people have encountered and how have they overcome it? Right. So here, this is how it starts. I'd shown you pentamidine. Here's netropsin. It's a natural product made by bacteria to kill other bacteria or to keep other bacteria from growing, cytostatic and to some degree cytotoxic. This molecule binds the DNA in this way. So it is single rather than what I have been showing you so far. These are two of those types of molecules stacked on each other. Like here's one chain, here's the other chain. But the natural product which inspired that sort of design is this. So it has two rings attached with the amide bond. And distamycin, which is a close relative, has three perols, N-methyl perol attached with amide bond, right? So imagine three of them, and that is the natural product distamycin. And the cool thing about this is, of course, it sits in a narrow minor groove. So you can see the DNA here. This is your major groove. You can see how much space there is here. This is your minor groove, right? On the other side of the major groove. And it is narrower, right? You can see that this space is less than that space. Now, most proteins that our bodies use to turn the genome on, off, or control it, like this sort of space, because they use a alpha helix, a protein alpha helix, that fits nicely in this space right here, like that. That a helix can dock beautifully, lying along the length of the major groove. Small molecules, however, have limited surface area and require a narrower pocket, if you will, right? So the minor groove offers the opportunity to dock one of these natural products, and it likes natural product sequences that allow it to dig as deeply as possible. So base pairs like A's and T's that don't have anything pointing in the minor groove, right? Imagine, here's your minor groove right here. This pocket here, if the bottom is flat, the molecule can come as close as it can to the base and use all the interaction surface it can get in the minor groove. And that's what's happening. Here are the phosphates. You can see these oxygens and phosphates right there, sorry, phos. And so It's almost at the edge of where the molecule is. So this methyl is sticking out a little bit right there. This N-methyl is right there and right there. But basically, the molecule has completely burrowed in. And what this led people, for 20 years, people studied how these molecules, distamycin, natropsin, other minor groove binders bind. And based on that, they started making changes to the edges of the molecule. So let me come back and say they started making changes to this side, which is closely approaching the bottom of the DNA in the minor groove, the closest thing that comes to the edge of the base. And so, based on that, people started generating molecules like this. So if you look carefully, you will find that distamycin core or netropsin core, right? That's netropsin and that's distamycin. The ends, of course, are very different. Distamycin, netropsin have guanidinial ends and amidine ends, etc. Here, of course, we don't have that. So almost 20 years of work from Peter Derwin and a variety of other people, Andrew Wong, Bill Lown, Dickerson, David Wemmer, etc. A lot of people across the world looking at the natural product netropsin, distamycin, seeing how it docks in the minor groove, and seeing how it prefers these flat 80 base steps, right? People started changing the edges of the small molecule. So here's a ring. Instead of imagine putting a hydroxyl there. When you do that, all of a sudden, the hydroxypyrrole prefers to bind a T, even though it would sit more happily in a AT or a TA base step than a imidazole. So now you replace a carbon with a nitrogen, and that nitrogen lone pair likes to interact with the exocyclic amine that is sticking out in the minor groove, right? So in this case, with a hydroxypyrrole or a pyrrole, having the exocyclic amine sitting out in a minor groove would be a problem, which is like a thumb. Or let's say instead of having something like this, there's something like that sticking out in the minor groove. Here's that amine sticking out the guanine. How do you dock into that? Right? Replacing that carbon with a nitrogen in this imidazole ring, you utilize that engagement. You form bonds with it. And so by replacing rings one by one using amide coupling, they were able to test thousands of different rings and come up with this. At least hundreds. Dale Boger at Scripps tested at least a library of a couple of hundred molecules, rings, to find that this combination perhaps works really well. And what you'll also notice is that what Dervin's main contribution was when they did the crystal structure, here's a crystal structure. Um, what they found is that the molecule that they had made, so these are pyrroles, meaning these types of rings, two of them in a row, and then they have a hydroxypyrrole, this one here, and then they have an imidazole right there. So these four rings right here, right? They made that molecule, and when they did the structure, they found that two of them were stacked on top of each other in the anti-parallel way in the minor groove, right? So you can see that this molecule is the same as this molecule, except rotated around and bound the other way. And when they found that this is the way the molecule likes to bind at a certain concentration, right, they were getting high specificity. So having two rings stacked against each other, the base step that the molecule is recognizing, it would do so really well. But if you didn't tether them, these rings could be in different registers. They could be sliding up and down. You could imagine that this ring shifts over by one, right? So the imidazole is here. Now this one will recognize A or T. This imidazole stacked against that pyrrole will recognize GC. Instead of going CG here, it might do CG one base step over, and you have no control. And individually, these things might bind by themselves in other places. So how do you make sure that you lock in this arrangement, that this ring and that ring are always next to each other, sitting on top of each other, right? How do you do that? So they tried different linkers and something as simple as what connection to put here to connect these two together. Like, imagine a linker attaching these two, right? If you have too long a linker, the molecules can still shift. If you have too short a linker, the molecules do not dock as well. And what type of linker should be an aliphatic linker? Should be a peg linker? What would you add? All of this took surprisingly long, but when they did find the linker, and it required 3 to 4 carbons, and that's it, that distance, right? Then they were able to lock in ring stacking of this kind. And when they got rings stacking against each other and digging into the minor groove, they were able to get specificity. You'll notice that this is almost 25 years ago, right? And yet, these things have gone to clinic and sort of struggled a little bit, and I'll explain why. So, anyway, as soon as they had this, they decided to ask, can we use them as synthetic regulators of genes, right? So, this base pairing. Oh, I don't progress. And so this is where they made essentially synthetic genome readers, small molecules that can recognize or read the genome sequence, right? Once you have this, what also happens is since you've paid the entropic penalty, right, by tethering these two together, these molecules now show nanomolar affinity for their sites. So this is incredible. It's almost like a protein that is binding in a major groove. And the important piece here between 98 and 2000 was that people did not think that such sort of molecules that only bind, let's say, 6 to 7 base pairs, maybe 8 base pairs, could ever be sequence selective the way our proteins are. When they go to the genome and look for the binding site, they show exquisite selectivity. Or so it was thought 25 years ago. So we at this point started our lab and asked, how good are these? But before I go there, let me show you what, once they had this key, what all they were able to do with it. So starting with this simple natural product, I showed you a hairpin polyamide, and this is the architecture we will use, and I'll point to it. But you can also have things bound in a linear way, like this, one to one. One stack of rings, a set of rings, bound in the minor groove and reading one base step at a time, right? And so this is what happened over the next 15 to 18 years, where people started using these types of rings. We just talked about N-methylperols, N-methyl imidazoles, chlorothiophene. We've talked about hydroxyperols, and also fused ring systems like this. So using all sorts of things in place of N-methylperol, people explored what type of specificity can we get, right? And you can see an example of what I just showed you, where you have N-methylperol, N-methylperol, imidazole, etc., but also a chlorothiophenes, and they found this actually prefers a T. So this is the nomenclature typically people are using nowadays, where N-methylperols are shown as an open circle, N-methylimidazoles is a closed circle. So now once we've done that, now you can go up here and see how people have tried to make different architectures, if you will, different types of arrangements. You can have long linear molecules. So this here, right, or rather this here, is no different than these two things, two rings, perol rings, like that. Okay? But now you put a beta alanine, a linker, if you will, that allows you to trace the molecule around the minor groove, which is turning as well. So you essentially are putting a chemical hinge, and then you attach another set of rings, if you will, two rings, hinge, rings, etc. And this is sort of overlapping a little bit. This is overlapping completely, like the structure I just showed you. People have tried to make cycles so that there's no chance that the molecules will open. We connect them both. You try different rings, etc. Right? And also the turn here, as you can see, a variety of different things have been tried to try and see if we can make this more rigid so the molecule does not have a chance of opening out or and binding DNA in this format, right? Because at the end of the day, these turns essentially are flexible tethers as well, no different than a beta alanine, maybe perhaps even more permissive. But it is the ability of the rings to stack each other and the energy they get from that that allows them to adopt this conformation. So a lot of energy has gone into making rings more stereo specific, making them more rigid. As you'll see, some of these turns have a fixed, actually, I do not have that example, but some of them have actually a rigid turn. The arrow ring is used to connect the two hearts. And more interestingly, at this end of the molecule, people have attached all sorts of things, payloads like uh, adamantine containing molecules. We have attached peptides, this sort of dipeptide. People have attached steroids. And people have attached histone deacetylase inhibitors, among other things that have been attached at this end to see if we can, once we get to a location, bring something that would otherwise be toxic to that site and make it more effective. Right? So this is what has happened since the first codes were described, right? And it has been quite effective. And yet it has not made it to clinic. So we, around this time, as you can see, I had started my lab, started asking why is it that these molecules that show such specificity, right? I mean, I'll go back one more slide. This such specificity that people are calling them base pair reading rules, recognition rules, where you can design a pair of rings and you know that they will like CG. You flip the order. Here's the metazole parole. You can see it's exactly flipped, like this essentially. This thing will recognize C on one base step and G on the other, G on this base step, C on the other, right? So if you're just reading this strand, if you will, it would read C, A, T, G, okay? And yet, when you have that kind of specificity and you have all these interesting designs, when you go into cells, it doesn't work so well. What is the problem? So roughly at this time, we said, how good are these recognition rules? When people study the molecule, right? They do it for the site that they think it should bind, right? You've come to this problem with rules in mind. So, well, okay, this thing should recognize a G, G, A, something, right? But does it recognize only that, or can it recognize related sequences? Could it go G, T, A, G, A, T, G, T, T, G, A, etc.? Right? By the time you do this for something that binds 8 or 9 base pairs, you're up to 32,000 different sequences. As chemists and biochemists, molecular biologists, very hard to measure binding to every single possible site. Maybe the molecules have new specificity that we had not expected. So what we set out to do is we put all 32,000 sites separated from each other on a glass slide. This is way back 20 years ago when this technology was brand new, right? And we were able to make slides like this and put a fluorophore, a dye, organic dye on the molecule, and ask if I gave you every possible sequence, every possible 9-mer, every possible 10-mer, 10-mer is about 500,000 different sequences, 500,000 sequences, different sequence. Um, and what would the molecule like? And when we did that sort of work, we were able to see most of the binding sites. So this is a number of sequences. Most of the binding sites are not bound all that well. And very few, virtually impossible to see. A few hundreds are bound by the molecule that we designed. The same turned out to be true for proteins. So we took proteins and we put them on the array also, and we saw same type of profile. Majority, so if you have 500,000 sequences, maybe 450,000 are here in the noise. And 50,000 now are spread, maybe looks a little bit like that, little more like that. And very few, maybe 40, 50, 100 look like this. And that is what the small molecule, the protein like to bind. But as you made protein as molecules bigger, or when you look at multiple proteins, now you needed something that was much more complex. And so as sequencing technologies became more powerful, where you could sequence hundreds of millions of sequences, we switched from doing arrays to sequences. And when you did that, again, the answer turned out to be like this. Few sequences are bound very, very tightly, both by small molecules and by proteins. But there are many related sequences that are bound to some degree. And if you take all these sequences, take examples of these sequences and say, how well do they bind? Here's the affinity. You find that it's quite proportional to the way they bind on the arrays. And what is even more interesting is that it is these kinds of medium binders, weak binders, that you find in the genome as being important for proteins to do their job. This is a Gata factor. It controls how your heart develops. And if there are mutations that it cannot bind this sequence well, you have missing valves, you have missing components or smaller components of your heart, and major cardiac defects growing up, right? And so it is not the best binders, but the moderate to weak binders that are important. So we stopped looking at things in this way and started looking at things in this way, where everything that is related to this sequence is in the middle. So it is, imagine GGATAA, that would be here. AGATAA would be somewhere here. If you make a change in the back end, you have AAAA, that is probably somewhere here or there, right? And so what is around your site, but the core site is the same. That is, all of those guys are here. The first string out is if you have a difference at the first position at any one position. So right there, you can see in this section here, actually, where my pointer is, if you remove this G and you put A, C, or T, binding goes away completely. But you can change these base steps and still have binding. You can change this A here and still have reasonable binding. So things like this, we start looking at as energy landscapes. Where on this affinity spectrum, where on this binding energy spectrum are you? And if we can pick this one out, how might we target? This is the way we started thinking about the genome and saying only the sequence now. In the genome, you can bind all these things depending how frequent they are. And when we did that, we found that the design molecule, like this one, should bind G, G, A or T, A or T, meaning W, because this cannot, especially this one, cannot tell the difference. And you can see that T and A equally represented. But you look at all the proteins that make us. Here's the Gata factor I was just telling you about. Here's NKS 2.5. These two together, important for heart development. And one of my colleagues, Jose Rodriguez Martinez, is professor there at UPR. He worked on these proteins, and as I think continues to work on cardiac specific transcription factors. And you can see how they're binding. They're putting a helix in the major groove, and they're reading the edges of the bases. And so if you look at the actual number of base pairs and the amount of energy or information that the protein is using to recognize this site, you'll find that it is not that different than what the small molecules recognize. About 7 base pairs, some of those base steps are more important, as you can see here, than others on the other side, right? So once we had this, we were sure that we could use these molecules to target genes. And at the time, people started using these kinds of molecules for various diseases. So here's a cancer. I believe this is prostate cancer. You can see the prostate cancer. And started making molecules that will interfere, meaning they will compete. Can we make a molecule that will bind on this minor groove and push this finger out, push this native protein? So once it is bound, the protein cannot bind because your compound is docked out there, right? Can we do that kind of competition? And that is how we control. We just keep the transcription factor that may be driving cancer, something like P53 is here somewhere. That's P53. Important in 70% of cancers. MYC is another one that can bind a variety of sequences. Can we keep them from doing their job and reduce cancer that way? So this is what they tried for prostate cancer, for glucocorticoid receptor. It binds a sequence. You make a molecule that like the same sequence, and if it binds there, it'll keep GR from working, and now you don't have genes being turned on. In other examples, where a different transcription factor with different sequences brings GR, the molecule will not compete. So it'll be very specific for the size you designed it for and not be toxic like most therapeutics that I first showed you in slide 1. And they showed that you can reduce prostate cancer levels, etc. But this is effective in cancer where you can give very high doses, and if they're off-site targets or off-site effects, side effects, you can tolerate it because that is a solution. But how do you use it broadly? Right? How do you use it chronically, all the time, every day? If you were to use it, what would be the cost? Pretty high. So what were the challenges? Why did not it become most widely used? So here's what um, people had done in the literature. They had taken a molecule, a hairpin polyamide, we call it a hairpin because it is termed this way, that targets a binding site for a transcription factor that is important for bringing blood vessels to cancer cells, to tumors, right? HIF1 alpha. And it's involved in regulating genes like VEGF, which provide growth signals to capillaries that are endothelial cells that are coming towards the tumor. And so they made the molecule PA4, polyamide 4, to bind the binding sites of HIF1 alpha. It's called HIF1 responsive element. If the molecule binds here, it should actually push the protein away. I don't have the exact cartoon, but the point is that it should compete, binds here, and should compete the protein away. And if it does that, the gene should be switched off. And it is switched off a little bit. So this is log twofold, which is about 2 to 3-fold uh, down regulation of the gene. But in that same data set, they found another gene, endothelin, also involved in blood cell development, down several fold, much more effective. And there, the binding site is not as strong. You can see this is on the orange yellow scale. Here it is in deep blue. So if I were to go back in this energy landscape, this would be almost at background levels, at this level. You know, but the difference between this gene, VEGF, and endothelin 2 is that there are many weak binding sites all across the gene, especially around the start site also. So could it be that together these weak binding sites, many small low affinity binding sites, are together somehow keeping this gene from turning on? So initially, this was not very, positively looked at. People thought that this is not going to be the way things work. So we had to go do an experiment. And the experiment that the student did was to explore the specificity of that molecule in live cells. Now, the trouble is, how do you do that? The molecule will shift the minute you break cells. So what he decided to do is put a sorrelin. This is the intercalator that we've used that can cross-link to DNA with UV light, and affinity handle biotin, right? And let me just show you the molecule rather than wait. He took different types of hairpins or linear molecules, putting sorrelin and biotin at the end. And biotin allows you to pull the compound out again. But this time, what you've done is that after you've incubated the compound with cells, these are human stem cells, incubated them, you can see the little compounds there. Then you cross-linked with light. And what sorrelin does is it's sitting between base steps and you hit it with light and it'll form these adducts. It'll form covalent adducts with both strands, right? So you form the adduct, you break the genome, and you use the biotin right here to pull out the small molecule DNA construct. So here you are, you're pulling this guy out, you're pulling this guy out, everything else you're removing using streptavidin, which can bind to biotin right now. Once you pulled it out, you reverse the cross-link. You take the DNA and you ask, what is the sequence and where in the genome is it? So Graham called this cosmic. And when we did this, we found that the molecules are like this hairpin here, are bound to places in the genome. And because we had done binding in vitro, this is inside a cell, this is in vitro, we knew what it likes to bind. So we went back and said, based on the different sequences you like to bind, what kinds of sequences are underneath this peak inside the cell? And when you look at it, there's some very strong binding sites, some moderate binding sites, some weak binding sites. And this is what the sequence looks like, right there in the middle. Another molecule, this linear molecule that targets GAA, 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 also shows these peaks. And nothing here, right? Very small sum. So if you go back and say, what do you like to bind in vitro? You find underneath this peak, there are only moderate sites. This really strong site for which everybody for 25 years, people were trying to design molecules that go here. There's barely any binding. The strongest site is barely bound, multiple weak sites. And you can see the sequence here, GA stretches, right? And this thing likes GA, GA, GA. So when you have these sorts of sequences, when you have many of them, that is where the compound likes to sit. And this is 6, 7 years ago, almost 10 years now, when we realized this, right? That the way this molecule or this molecule, and here it is supposed to target TACG, and you can sort of see that sort of sequence. If you look at it carefully, you can see TACG here. You can see TACG there, but you can also see a related sequence, TACAG. So it is the range of sequences, something in this affinity range, but many potential binding sites is the reason why the molecule is enriching here. You see that? So in vitro, this molecule will bind this sequence. In cells, it can bind all these things because they're next to each other and they are low energy wells. So you can say you can get stuck in local minima. You have a lot of these sequences, a couple of these, and the compound comes along and then is trapped there in a living cell. If you just make one site for this kind of compound, it can bind there, but the cell is a living thing and it is pushing these molecules off. It is only when you have multiple binding sites do these molecules stay. So this was the change in our thinking that allowed us to do all sorts of things, and partly why I'm at this conference today. So this is the understanding. The compound can bind DNA, naked DNA, or DNA on a nucleosome, packaged in these nucleosomes. Nucleosomes can stack on each other, and you can have multiple open binding sites, like this one is facing solvent. And it is a collection of binding sites. That is where the molecule will spend a lot of time. It's a collection of binding sites on straight DNA or in compacted DNA. That is far more important than any one single tight binding site. Okay, so this is where we were 7 or 8 years ago, where we knew how to make molecules to target cis. This is a contribution of many, many, many people over the years. Now we understood how the molecules access the genome in living cells, and they like to bind multiple binding sites. So we were using not just a motif, but the full range of binding sites. And then when we sum the binding sites in a given stretch of genome, we can more effectively predict where the molecule is going to be in a living cell. So this is a major change. And from there, then we started developing molecules that target disease genomes, where these sequences occur in large numbers. I'm going to switch now and let's see how much time we have. We have about an hour, is that correct?
We have about 35 minutes.
Sorry?
We have about 35 minutes remaining.
35 minutes. Okay. So I'll go through this somewhat quickly and then we'll leave time for questions. So second half, if you guys are not on board yet, I've brought you to this point where this is the way we think about DNA targeting, genome targeting in living cells. Now, how do we go from there to do something to the genome where you want to turn on or turn off the gene, right? So this is when in parallel, this idea of chemical induced proximity, bringing things together using small molecules, came into popular view. We had been doing this for a long time, almost as long as Protax, earlier, in fact. Um, but it became effective in different formats and it just took off. And I'll show you the history of chemical induced proximity. Proximity in biology is absolutely important. So what we are really doing is we are using this biological principle of bringing things together. Here's a cell, things distributed all over, some of them in small condensates where they'll spend their entire lives in these little vesicles, or these condensates where molecules come together. But here's what's happening on the cell surface. You have receptors that are usually hanging out as monomers, excuse me. When a signal comes along, like you have to grow, or insulin, or growth factors, or inhibitory factors, or even recognition between cells, right? This is T cell receptor interacting with a target cell. These monomers come together and they're held together. And when they come together, this part of the molecule that is facing inside the cell, they engage and then they turn on a cascade, a signal transduction cascade that tells the cell, grow, don't grow, stop, you're in contact with a neighboring cell, etc. All those signals, the receptors are there and they're distributed all over. But if you now start engaging them and bringing them together, you turn on a signaling event. Same thing happens with proteins that are done or are misfolded or somehow damaged. How do you get rid of that? You have a whole system that engages the protein with machines that will ubiquitate it, tag it. These are E3 ligases. This is the adapter that will talk to the damaged protein or a target protein. And once these two are brought together, this enzyme can start tagging it with ubiquitin. And that tagged version then is recognized by this barrel-like machine called the proteasome that Jose studies in the lab. It can bind these ubiquitals, this lid region, open up the protein and feed it inside the barrel, and out comes fragments, broken down pieces. Here's proximity. If these two interactions are perturbed, this protein will be stable. If these two interactions are forced, even a perfectly fine protein is tagged and degraded. So this is at the surface, this is in the cytoplasm. And what we focus on is what is going on inside the nucleus. So here's your nucleus. And here, your red blood cells, people who have sickle cell or other diseases, right? Proximity is absolutely important. This is a control region. It's called the locus control region, LCR. It has a lot of proteins bound. If it chooses when you are a fetus, before you're born, this gene to make blood is made. It's hemoglobin beta. Delta, sorry. And this fetal hemoglobin has higher affinity for oxygen. So it can extract oxygen from the mother's blood, right? Once you're born, this locus control region shifts a few thousand base pairs, even though it's not to scale. It is choosing between this and that. The proximity of the proteins bound at the locus control to this gene or that gene controls the switch from fetal hemoglobin to adult hemoglobin, right? So this sort of proximity, what controller is brought next to what gene, turns out to be absolutely important how the genes are being turned on. And so people in the chemistry world have learned this lesson and designed molecules to bring degraders together. This is extremely powerful these days. We've been doing this for almost as longer. And more recently, people have been using these sorts of smaller molecules to transport things into nuclei, bring different types of proteins together, etc., even bring receptors together. So these are the ways induced proximity is being used, and I'll show you the example of a power of this. So this is a classic way of making drugs. Since we're talking about chemical biology, this is a 100-year history of finding drugs by major pharma. It finds a small molecule that is usually under 500 Daltons, has a certain number of hydrogen bond donors, acceptors, a certain solubility properties. These molecules will bind to the protein that you're trying to switch off in a stoichiometric way, right? And usually in a non-covalent way, but now more recently people have been making covalent molecules that'll engage. In any case, it'll target most of your proteins and may target undesired proteins, but it has to be stoichiometric, has to be specific, has to have really high affinity, right? And what now you have to do is that this is one way to do it. And if there's any change, if cells have a mutation, this drug is no longer useful. If you go too high, you might start hitting proteins that are important. So people started to imagine that could you take something that binds to your protein of interest, either as a unimolecular glue, if you will, something that bridges two proteins together, either just by using different chemical edges and the cooperativity between interacting partners, or simply you have a strong ligand, a ligand like this, attached to a second ligand that binds to the degrading machinery right there. And now what you do is you mark the interesting protein of interest with ubiquitin and lead to its degradation. And this is a catalytic event. You don't have to have a high dose of inhibitor to block every single protein. All you need to do is have enough of the degrader to be able to keep destroying the protein that you're trying to target catalytically, right? And this allows you to also engage proteins that may not have very strong binders, that may not have very selective binders, because the ability to ubiquitinate is important in terms of how things are positioned. And so now you're finding molecules that are not that specific being converted into bifunctional or even unimolecular structures that can engage the degradation machinery and lead to ubiquitination and degradation. So this has been used and has been so effective that now you're beginning to see very rapidly an increase in the number of papers and an increase of number of things that are being tested in the clinic for both uh, bifunctional PROTACs. So here's induced proximity, where you're bringing protein of interest to the degradation machinery with a small molecule, either as a bifunctional molecule or as a single molecule that can span the two, right? And so that's what's happening. And with that, you're seeing a launch of many companies. And in fact, Arvinas, this company here started by my friend Craig Cruz, has just had a drug that is being approved for prostate cancer. And they're now building many, many more. So this is where induced proximity has taken the world in terms of creating new types of drugs for proteins that are impossible to target, proteins that perhaps control genes. Here's an example of a molecular glue, this type of molecule, that has been used to turn on genes. Again, using the idea of degradation. Here's your sickle cell, and as I was just telling you, In sickle cell, if there's a mutation in the adult hemoglobin, hemoglobin is not made so well. And as the locus control region changes, here's proximity going from the fetal hemoglobin to the adult hemoglobin. Now you start making this disease. If you have a mutation in globin, you create um, these types of cells, and this is a problem, and there are no cures. Now, what happens is if you can turn on fetal hemoglobin, even in adults, if you say, okay, don't make that disease gene, make this one, make a little bit of it, you can actually overcome sickle cell disease. And so people have been trying very hard using genetic engineering to try and either replace the gene or to shut down this disease gene and just make this one or make a fixed adult hemoglobin. But it turns out not to be that effective. The company that was doing this gave up eventually. Even though they could fix the problem, they could tell the locus control region to make gamma globin, or they could correct the disease version. The disease is prevalent in parts of the world that you cannot pay $2 to $3 million to fix the problem. So it's absolutely important to still have small molecules, and that's where a colleague of ours decided to make a degrader. All these things are controlling how the locus control region is going to turn this gene on or that gene on or the adult. So if you can switch off any of these proteins, degrade any of these proteins, the idea is that it'll change how this locus control region talks to these two genes. And so what they focused on is this one. They went looking for a glue and they found a degrader, a molecule that can take WIS1 and destroy it. And when it destroys it, the locus control region chooses this gene, and that is what is made. And it's now being developed as a therapeutic. Um, Basically, it is reversing the switch for sickle cell. And this is most likely going to be the one that is going to be used widely. Same idea of induced proximity, we were in parallel developing to turn genes on. So again, we go to how transcription natural systems work. These proteins can recognize DNA. This is some of the examples I'm showing you with P53, with GATA, with NKX 2.5. These are transcription factors that recognize specific sites upstream of genes that they're trying to control, and they bring machinery to that gene, right? They move the nucleosome around, they bring polymerase that can now bind the start site. It'll start making RNA. Transcription factors can also bring proteins that allow it to get through nucleosomes all the way to the end, right? So that is what this protein can do. If you look at the anatomy, what are the pieces of a transcription factor? This is what they typically have. They have a region that will bind DNA. They have all sorts of other pieces that can respond to different signals, like nuclear receptors can respond to estrogen or androgen or glucocorticoids, etc. So some signaling component. And then a regulatory domain, a domain that can bring all these machines. So this part of the transcription factor that can bring machines that move nucleosomes, polymerase that can transcribe, enzymes that can modify polymerase so it can get through nucleosomes, etc. All that is encompassed typically in a regulatory domain. You can separate everything out and just take the basic pieces and paste them together. In fact, even replace the DNA binding domain with the kinds of molecules I was just showing you, hairpin polyamides, right? And then make a molecule that is functional. Here's an example from a cartoon to actual structure. This is one transcription factor, this is another transcription factor, and they both talk through this short peptide. One is bound to this groove, the other one is bound to this groove, and between them, the connection through this linker, parts of it are not defined, but the other end of this protein is interacting. So they're both interacting cooperatively, right? And what we did is we removed this protein entirely with a molecule that can read this minor groove right there. And off that, we now display this peptide. So here's induced proximity. The compound will bind, polyamide will bind a sequence. It presents a ligand that can engage a protein of interest. And together, now using this transcription factor, you get its regulatory domain. So this is just a part that is DNA binding. But attached to this is the regulatory domain of this factor. So now, through a small molecule or a synthetic molecule, it's no longer small. It is 3 times the size of a typical drug. This would be a typical drug, about 500 Daltons. This would be a typical drug, but together it is about 3 times the size of a typical drug. But things like this can bind in DNA selectively, recruit a partner, and use that to drive gene expression, right? And so we applied that to a disease in Friedreich's ataxia, where we thought, can we use a polyamide to target GAA repeats? I just showed you we have a compound that targets GAA repeats. Through a linker, can we present a second molecule that'll bring machinery that allows transcription, right? We chose Friedreich's ataxia. It's a disease that is based on one single protein called frataxin, which is an iron chaperone, allows the assembly of proteins that make ATP inside the mitochondria. Encoded in the nucleus, translated in cytoplasm, transported to mitochondria, and there it functions as an iron chaperone. If it goes down, people develop first spondylitis and then all sorts of walking disorders. Therefore, it's called ataxia. And then eventually cardio morbidity because the heart is not working so well. There's a muscle that needs to work all the time. If it can't make ATP, it stops working. And that's how people tend to die. So what happens in the disease is that this is the normal gene that most of us have. It's called frataxin. And in the middle of the gene are these sequences, GAA repeats, that are expanded in a diseased individual. And that expansion, right, keeps the machinery from traveling. And so when this gene cannot be transcribed because there's this roadblock in between, the level of mRNA goes down, and you can see that here, right? This is a normal person, this is a patient. And there are lots of reasons why GAA repeats might interfere with the way polymerase can travel. Maybe they form unusual structures, maybe they get wrapped up in huge chromatin. But the question is that you're still making a little bit, and how do you do that? Right? So some polymerase must engage, some of it must get through. And so a student in the lab went and asked, where is the polymerase? And the polymerase is binding start site just fine. It can even start to transcribe, but then as it gets close to the GA repeats right here, it stops. You can see a lot of polymerase down to nothing. Very little is getting through. You can see a little bit down, all the way down to the G. And this is what's keeping these people alive, because you need frataxin to make ATP and ATP function. So we decided to say, what can we make that will allow, based on natural transcription factors, can we use the induced proximity, make a bifunctional molecule that'll bring machinery that allows polymerase to get through, just like in the natural system, if I may, too many missing pieces. This transcription factor can bring this machinery to allow polymerase to get through nucleosomal hurdles all the way to the end. Can we bring this kind of machinery to the polymerase that is stuck right there, right? And so using induced proximity, 2 modules, one to recognize GA in the DNA, right? It can bind GA. And then the second ligand, interaction domain, that can bring in elongation machinery to take this polymerase through the blue repeats, right? If it talks to the polymerase, we should be able to make RNA. And so what to use for GA targeting, I have already shown you this molecule. I've already shown you that it likes to bind in cells to the repeats, right? So we already had this part worked out. Now on the other side, what to use? And so here, we focused on what was known in the literature. Transcription factors bring in this kind of machinery, elongation machinery, transcription elongation factor components, right? Different things. And those things work on polymerase, right? This was already known at 10, 15 years ago. Right? And so in cancer, factors like MYC do this a lot and make a lot of genes that tell the cells to continue to grow, right? And so how do you prevent this from happening? People had looked for ligands that can keep the elongation machinery from binding. Even though transcription factor is bringing it, it cannot associate with the polymerase or cannot associate with local chromatin. And so they had found this molecule called JQ1. In our situation, the reverse is true. The polymerase is there, but it cannot get through. So we need to bring this machinery. Instead of pushing it away, we need to bring this machinery. And so we thought, why not use JQ1? This is physical chemistry, right? What if you were to bring JQ1 to repeats? Could it engage machinery, right? Instead of displacing it by competitive inhibition, could you recruit it? And if you brought it, would it provide all the signals that this stalled polymerase needs to be able to get through? And so that is what we did. We made a molecule that binds GAA, linker, and attach JQ1. And what we saw is that it can turn on genes. It can turn on frataxin. So this is actually in three different patients. I think I may have flipped the slides, I'm sorry. But in three different patients, we were working with Novartis, where they took the cells, they made them into induced pluripotent cells, and then converted them to neurons. So you can see this part of the cerebellum is now being rapidly atrophying, and that is what leads to ataxia. They took patient cells, neurons, and what you can see is that if you have the compound, that bifunctional compound that can bring elongation machinery, you start making that protein again. Nothing normal is changing. This is beta actin, but the frataxin gene is now being made, and this allows patients to function. And then we tested it in, so that is where you've taken cells from patients, converted them to pluripotent cells, and then made them into neurons. And then I had colleagues who went directly to patients because they were trying to genotype patients, figure out what kind of ataxia they have. They would take blood cells and they would use some for genotyping and find out, oh, well, in the frataxin, this person had so many GA repeats. The other half of the cells, we told them instead of throwing them away, can you please treat them with our compound? And what you can see in different patients, this molecule is able to turn the gene back on. And if the patient is making a lot of frataxin, more than half, the effect is small. If the patient is making very little, so all that is normalized to one, the gene is made to a larger degree. But if you take all these numbers, it is essentially restoring, bringing back to normal rather than making a lot. It is bringing back to normal the amount of frataxin the cell needs. And so, what we think we have done, this is all the drugs people were using before to deal with the disease. And what we think we've done is by going and developing a molecule that targets GA repeats, genome targeted, induced proximity, bringing the missing components to that site in the genome, we've been able to fix, hopefully, a disease that until now had no cure. And so it's new mechanism. We started a company to optimize it to now take it into patients. And so this is what we think we have achieved. We've made a bifunctional molecule that can target, bring in the missing pieces, and inform the polymerase that yes, you can get through because it's associated with proteins that allow it to get through. And simple molecule, bifunctional molecule of this kind. And that is where induced proximity has taken us now. And we are now applying it to all these diseases where different repeats are happening. So the company is doing some of this. In the lab, we are thinking of now going after a couple other diseases where people are interested. So Huntington's is one of those. And I'm at a Huntington's conference, and they're very interested in taking CAG repeats many, many times in the Huntington gene that is causing to neuronal failure, neuromuscular failure. And so can we build molecules that will target CAG repeats, and in this case, turn the gene off? In Friedreich's ataxia, we were turning the gene on. In Huntington's, you're trying to turn the gene off, right? In muscular dystrophy, CTG repeats at the back end of the gene. So this is the start of the gene. Friedreich's ataxia happens because of GA repeats in the intron. Huntington's, it is in the coding region that you have CAGs. Now, if you shut down that, you won't have the disease, or so the expectation is. In Ewing sarcoma, this type of repeat happens in the green region. Many, many, many repeats allow different proteins to bind and turn on genes that give you this type of lesion, etc. So these are some of them. There are about 70 different diseases where we can use the idea of induced proximity, things, molecules of this kind, want to recognize the repeat. This is GAA, GAA. You could imagine CAG, CAG, or CTG. Attached, instead of JQ1, which is bringing the elongation machinery, attached to things that will inhibit transcription or will modulate post-transcriptional processing, where you have the gene is made, you need the gene, right? What you don't want is the tail that has these repeats in it, which is causing the disease. So the RNA, very hard to drug RNA, but perhaps you can keep it from being made at this stage. So that is where the field is going. In the meanwhile, people have attached all sorts of molecules and tried them. Here's a steroid, here's a peptoid, here's an HDAC inhibitor, etc., to try and see if we can make these bifunctional molecules to target unique sequences like 13 mers in this case, cancer cells, in this case, HIF and alpha binding sites, and other um, hijacking natural transcription factors as we have done. And so that's where that is heading. And it's beyond even trying to regulate transcription that way. People are now using natural transcription factors and building molecules of the kind that we had built, basically inspired by some of the stuff we've done, to try and see if we can regulate death genes. So some of these cancer-causing factors are on in cells. Can you now force them, instead of switching genes off that promote death, into things that switch on genes that drive cell death? So you only kill cancer cells and you don't kill any other genes because you're rewiring the regulator. In a cancer cell, these proteins are trying to keep death genes from being made. By building a small molecule that brings machinery that forces death genes to be made, you flip the switch. You're going from switch off to switch on, and now you have a therapeutic effect. So this is being developed. There are ways to bring machinery together. Can you bring receptors together? And instead of trying to do signaling this way, can you enforce signaling independent of induced proximity? So the people are trying to rewire. And here is an example, a review written recently by Lucia Ciuli, that talks about all kinds of bifunctional molecules and induced proximity that people are developing, including things that target RNA. Somewhere here's ribotax, if I can find it. Things that can remove marks, stabilize proteins, but at the heart of it is, can you bring two processes together using a small molecule and drive different types of uh, outcomes? So this is where the field is, and your generation is going to be doing a lot more of this. Just recently, a molecule that failed to degrade, a PROTAC that failed to degrade but brought proteins together, led to a drug that was sold for $3 billion with a B. A failed molecule led to a company called Halda Therapeutics, which was renamed Riptax and sold as a therapeutic for prostate cancer. So this is what we've covered today. There's a whole new world of targeting undruggables that I will not go into, but I present to you as covalent drugs that can bind things that don't form pockets, flexible peptides that have unique properties, condensates that can form these little balls in nuclei or in cells that are important for disease or important for function. People are going for things that were unimaginable just, until recently, right? Disordered regions, condensates, and making covalent molecules so that you don't have to have a really high affinity binder, but you can now have sufficient selectivity to target a ligand of interest. And with that, I will stop sharing and take any questions that you might have. I've gone too far.
Thank you.
So we touched a lot of things, and I know there was too much I was trying to present in a matter of an hour, 20 minutes, but I hope this was useful to give you a sample of where we are.
Right. Question you guys have? Or others?
Do I ask? Can you hear me?
Can you hear?
I can hear you faintly, yes.
Okay, so hi, I had a quick question. I don't know the name of the genome reader that you were showing throughout the whole presentation, where in the last couple of slides, you kind of shared the specific type of compound that had a lot of drugs incorporated to it. Do you know what I'm referring to?
Yeah, you're talking about the molecules that had little ligands attached to it, right? Like JQ1.
I don't know the name of it, but yes, one of the last ones that we were seeing, essentially a lot of drugs attached to the different readers. I was kind of like wondering from the perspective of a chemist, is this something that we can just find in the literature? Let's say that I'm working on a specific type of cancer and I want to see, oh, could I attach the drug that I'm making to an already found.
Absolutely. The answer is absolutely yes. So if you have a ligand that you have come up with or you have identified in literature, you can tether it to another ligand that may take this to the site that you want on the genome or to other proteins or even lipids. Absolutely. And the challenge then becomes until recently, people didn't, as chemists, medicinal chemists especially, pay much attention to linker lengths because you try to make it as short as possible. to have as small a molecule as possible, more drug-like that could be orally available. Um, that's where the focus now goes. And people are using AI to try and figure out how close do you need it if you already know the surface. If there's two positive charges, maybe you need a longer linker to keep them apart, but still engage them. So that's where the focus is going. But in terms of ligands that you may have found or you may have developed, and if you want to increase their function or see if they can be used to leverage induced proximity for other functions, absolutely. The other thing that people have been doing that I didn't talk about is they've been using things that'll use sugars or even other breakable bonds, metabolizable bonds, disulfides, etc., or esters, so that you can attach two or three different drugs that might work independently, but they're delivered in equal amounts into the cell. And once they go into the cell, the linker is broken and the drugs are released. And even prodrugs that they go into cells, but because of the neighboring piece, an enzyme comes and modifies your drug of interest, right? And in a way that now makes this drug really toxic that you wouldn't want otherwise, right?
I,
To be floating around in the cell.
I'm sorry, I seem to interrupt. I think she also meant about the polyamines, like if there's like a database or anything of like potential polyamines to target, like different genes. Uh, Correct me if I'm wrong. I think that's what you meant, like the specific polyamines for different genes and targeting them.
Yeah.
Yeah.
Yeah. So we are building that database. So if you're
Um,
interested, you should reach out to Jose.
do we have another question?
Yeah. Um, Thank you for the presentation. Now you mentioned drugging the genome and then the proteins directly, but I was wondering if you could use those genome readers in some way to then drug RNA. What would be the challenges of doing that? You mentioned that it was challenging to drug RNA, but yeah, I've seen a little bit of work done on that, like drugging the transcriptome. But I wanted to know a little bit more from your perspective.
Um, I'll tell you from the polyamide perspective, and then I'm going to ask Jose, our intrepid TA here, to tell you a little bit about drugging RNA with small molecules. So, polyamides, people have tried to alter them so that they would bind RNA, especially duplex RNA. So, for single-strand RNA, there isn't enough information or even interaction surface for these compounds to bind selectively. So, they don't bind RNA. Duplex RNA, because of the second hydroxyl, it um, and also the way the base pairs in RNA bind, it forms a more A-form shape, so the minor groove is more open and flat. So again, this class of molecules does not do so well binding that type of RNA. But people have tried to take molecules that do bind RNA and try to see if they can elaborate them, just like natropsin, the two-ring structure was elaborated into many, many rings to gain specificity. People have tried to do this with RNA as well. And then I'm going to ask Jose to step in. Jose, you might unmute yourself and talk about your experiences in terms of selectively targeting RNA.
Yeah. The problem with RNA is the high dynamic form structures that it can form. So RNA, people think about it just as a single strand, but it can actually form very diverse secondary and tertiary structures, but they're all very transient. So identifying ligands that can bind to those very specifically is very hard. Some people have claimed they do, but at the end of the day, it's very non-reproducible. I'm not going to get into that very like in
And
depth. But most of my work, and actually my lab, I just got a contact from my boss back in my PhD in Michigan. They found some interesting things with a screening platform that I helped develop to try and identify these types of interactions. So what I'm trying to say is that when people talk about targeting the transcriptome with small molecules, it's a taller task than what many think. But I think the main approach would be to just start targeting those secondary and tertiary structures rather than necessarily like with polyamines where we have like a defined minor groove that we can like kind of incorporate molecules into it. We need to still do a lot more research into structure probing the RNA before we can start talking about developing therapeutics for it, just because we don't understand the structures that very
so,
well.
yeah, I'll jump in again. And so this, you just heard Jose talk about 6 years of his PhD or 5 years of his PhD struggling with targeting RNA. They can form different shapes. They don't have the kind of druggable pockets. People are claiming that there are, and you also heard his pain when he said that I'm not going to talk about it because in the literature people have claimed that they can do this, and it has not been robust. The way people are targeting RNA these days, and again, you're seeing a lot of success in some a modality that had failed for many, many, many years, almost 2 decades, is antisense oligonucleotides. So people are using modified bases, modified nucleotides, right, that are stabilized and modified in a way to be able to get into cells and engage RNA in base pairing. So that sort of selectivity has taken the world by the storm. A lot of diseases are being targeted using that modality. And then some people are even trying to target RNA using proteins, small fragments of helices that are made from oligopeptide mimetics that are almost drug-like, within 600 or 700 Daltons, and then seeing if they can engage just enough to be able to perturb the state of the RNA. But those are the sorts of methods people are using to target RNA. The class of molecules we're working with don't seem to bind. And again, just like Jose, there are people out there um, who have reported that these things can bind RNA, and I don't want to go into it, but let me assure you they do not.
Well, thank you so much. We're going to give a round of applause to our guest lecturer.
Thank you, guys. And if you guys want to put any of this work, including connecting things to polyamides or targeting nucleic acids, let us know. And Aisha can tell you what it was like coming to the lab afterwards.
That's great. Thank you so much.
Thank you very much, guys. Thank you. It's a pleasure. Thank you, Dr. Tinako.